My dear students, I am Dr. Meera BK, working as Associate Professor in the Department of Zoology, Maharani Science College for Women, Bangalore, 1. In my last class, I have taught you what are known as molecular scissors or restriction endonucleases, which are indispensable for recombinant DNA technology experiments. In this online class pertaining to RDT, we shall learn few more other enzymes which find applications in this field. When I say there are molecular scissors meant to cut the DNA fragments, we should have some enzymes which do the opposite reaction, that is, which help in joining the DNA fragments. There we call them as molecular scissors. We should have something which serve as molecular glues. That means which ligate two fragments of DNA, and those enzymes which ligate two fragments of DNA are called DNA ligases. Now we shall learn about DNA ligases. Now, the enzymes which are used for joining two fragments of DNA, that is for ligation of DNA, are called DNA ligases. There are two sources from which the DNA ligases are extracted. First one, mostly you extract these enzymes from T4 bacteriophage after infecting E. coli cells. By now you must be knowing that phages are the viruses whose host cells are bacteria. So T4 bacteriophage is one such virus whose host is Escherichia coli. So once T4 phage infects E. coli cells, it starts producing the DNA ligases, and those DNA ligases are extracted. This is one source. And another source from which we can get DNA ligases is from uninfected E. coli cells, that is from Escherichia coli cells. Okay. So after knowing the source, we can have DNA ligases from T4 bacteriophage once they infect E. coli cells. Another one being from uninfected E. coli cells. What exactly ligation reaction does? Ligation or the joining or the gluing reaction essentially involves joining of a 5' phosphate group, 5' phosphate group in one fragment of DNA to the 3' hydroxyl group of the other fragment of DNA to form what is known as phosphodiester bond. Okay. To understand this, you should know a little bit of biochemical nature of DNA. I suppose you already know it in your previous semesters. Okay. So the ligation reaction does the joining of 5' phosphate group of one fragment of DNA to the 3' hydroxyl group of other fragment of DNA so as to form a phosphodiester bond. Now we shall study about both the types of DNA ligases. First one is bacteriophage T4 DNA ligase. To make the subject easier, I am just describing various steps pertaining to this enzyme under different subheadings. What exactly is the activity of phage T4 DNA ligase? What it does, it brings about ligation of cohesive DNA termini or NICs, ligation of cohesive DNA termini or nicks. What do I mean by a nick? A small cut in a DNA fragment is called nick. In Kannada, we say kachu. Exactly, the kachu is the nick. So, this enzyme joins the cohesive DNA termini, or it will join that nicks also. So, that small cut is removed and you will get a continuous piece of DNA. This is the activity of this particular enzyme. Then, for any enzyme, you have to give the substrate. What is the substrate? The substrate for this is double-stranded DNA molecule with complementary cohesive termini. Complementary cohesive termini to base pair to bring together 3' hydroxyl group and 5' phosphate group. That's all. So you have to give double-stranded DNA fragment with complementary cohesive termini. Then, how exactly the reaction proceeds? You can see the schematic diagram where I have represented a double-stranded DNA molecule with 5' end and 3' end. Now, here you can see, here is a guanine base with hydroxyl group and here is adenine with phosphate group. In the other strand, it is just the opposite. And now, if we take the cut DNA fragment like this, and if we give magnesium ions as cofactor and ATP, and this enzyme bacteriophage T4 DNA ligase, the enzyme catalyzes the reaction of ligation in presence of magnesium and ATP, finally resulting in joining the cuts. You will get a continuous strand of double-stranded DNA. This is what exactly happens after ligation. Okay? So, the activity is of this enzyme is to ligate like this by the establishment of a phosphodiester band, as I have already told you. Okay? And this enzyme not only brings about ligation of staggered ends. I have told you the meaning of staggered ends in my previous class. So, it is capable of ligating blunt ends also. So, as a substrate, you can give DNA pieces with staggered ends. You can also give DNA pieces with blunt ends or flush ends. Okay. Let us see the substrate for this activity where it brings about ligation of blunt ends. You should give a higher concentrations of blunt-ended double-stranded DNA. Blunt-ended double-stranded DNA probably which results when you treat the DNA piece with the restriction enzyme which brings about cuts so as to end up in blunt ends. Okay. Now, you should have one 5' phosphate group and 3' hydroxyl termini, that is a must, whatever be the reaction. You can see here, I have here the blunt ends, 2 pieces of DNA, and again I have to give or we have to provide magnesium ions, ATP, and also this enzyme. So, in the reaction, the ligation takes place by the formation of phosphodiester bond so as to get a continuous piece of DNA fragment. This is what exactly T4 bacteriophage DNA ligase does. This is all about T4 bacteriophage DNA ligase. Another DNA ligase is the one which is extracted from the uninfected E. coli. In the same sub-steps, I am going to describe the enzyme. What is the activity? Ligation of cohesive DNA termini or nicks. So, activity-wise, both the enzymes remain the same. Then substrate. What you have to give? Double-stranded DNA with complementary cohesive termini, that base pair to bring together 3' hydroxyl and 5' phosphate group by the establishment of a phosphodiester bond. And this enzyme is also capable of joining the nicked DNA also, the way the other enzyme does. Now you can look at the reaction here. I have 2 DNA pieces cut here. So for this also you have to give magnesium ions, ATP, and if you give E. coli DNA ligase, again DNA ligation takes place by the establishment of phosphodiester bond to end up in a continuous joined piece of DNA double-stranded segment. Thus, the enzyme catalyzes the formation of phosphodiester bond between a 5' phosphate and a 3' hydroxyl group into DNA fragments. So also it can join the nicked DNA. Now, normally this brings about ligation of staggered ends. It do ligase blunt end also. It do ligase blunt end, but you have to provide one more component for that action to take place. You have to give 10% PEG. 10% PEG. PEG stands for polyethylene glycol. Apart from magnesium and ATP, you have to give 10% PEG, or you can give phycol also, which serve as volume excluders. Okay? But if this reaction has to be catalyzed, in addition to this, you have to provide NAD plus for catalysis. This enzyme does not catalyze the ligation of RNA fragments. This is basically a DNA ligase. So students, till now I have taught you about 2 types of DNA ligases which we normally use in recombinant DNA experiments, technology experiments, which serve as molecular glues. Hope you have understood this. Next, apart from molecular scissors and molecular glues, we have so many other enzymes which are studied under the common heading nucleic acid modifying enzymes. Enzymes to modify the ends of DNA molecules which are needed based on the type of experiments you do in RDT. Okay? Now, as per your syllabus, you have to learn about only one such enzyme, that is alkaline phosphatase. Okay? Now, for the sake of modification of cut ends of DNA fragments and so many other fine tuning jobs that are essential in RDT experiments, we make use of several enzymes, hundreds of enzymes. What they do is, they supplement the cutting and ligating enzymes. Cutting enzyme is restriction endonucleases. Ligating enzyme is DNA ligases. So ultimately, the whole motto is to prepare types of ends that are required for DNA molecules which are participating in recombinant DNA technology procedures. One such important enzyme what we make use of, often make use of, is alkaline phosphatase. Alkaline phosphatase. We shall study the source of this enzyme. It is again extracted from bacterial cells. When we say bacterial alkaline phosphatase, if the source is bacterial cells, we say that alkaline phosphatase as bacterial alkaline phosphatase, BAP. Another source is you can extract it from calf intestinal cells. When we say, call it as calf intestinal phosphatase, CIP. Okay? These are the 2 sources for alkaline phosphatase. Now let us study the functions, what it does. Phosphatase, the name itself says, it removes the phosphate moiety at the 5' end of a DNA strand. It removes the phosphate moiety at the 5' end of a DNA strand, whether it is a staggered end, whether it is a blunt end, or a resist end of a DNA, double-stranded DNA, whatever be the case, it simply removes the phosphate moiety. Where it finds use? It is very much useful to prevent unwanted ligation of DNA molecules. In the next slide, I am going to show what exactly I meant. So, it is useful to prevent unwanted ligation of DNA molecules, which is a very frequent problem often encountered in cloning experiments. So, when linear vector DNA is treated with alkaline phosphatase, it does its job. That means it removes terminal phosphate, 5' terminal phosphate group. The moment you remove this phosphate group, this prevents recircularization and plasmid DNA dimer formation. I'll just show you the diagrammatic representation, you will understand better. So, the moment this recircularization is avoided, you can always insert the DNA of your choice through DNA ligase participation. So, this is the reason why we make use of alkaline phosphatase. Yeah. Students, look at this diagram which depicts the action of alkaline phosphatase to prevent the recircularization or religation of vector plasmid. See here at the top of the diagram, I have a plasmid DNA. What I have done is I have cut it because I want to insert the DNA of my choice in that place. The moment when I cut it with a restriction enzyme, I will end up with phosphate group and hydroxyl groups. Now, if I leave this cut molecule just like that, and if you give DNA ligase, so after some time, whatever I have done with lot of efforts, whole thing goes waste because recircularization of plasmid takes place. Then I will have no room to insert the DNA of my choice, and whole experiment is going to be a failure. Therefore, now what I will do is, after cutting the plasmid DNA with the restriction endonuclease, I will add alkaline phosphatase to it, either the BAP or the CYP. Which one to add when, I will tell you a little later. Now when I add alkaline phosphatase, the 5' phosphate groups are removed, so that I will end up on both the termini with hydroxyl groups only. I will have no phosphate groups, and under such circumstances, when I give DNA ligase, there is no recircularization of plasmid. So the cut ends stand naked, they don't join. Why I want this is because when it happens, when recircularization doesn't happen, I can always insert a foreign DNA segment of my choice in presence of DNA ligase, so that I will get a recombinant plasmid DNA molecule which I can transfect into the host cell. This is the purpose. I think by now you understood the significance of removal of phosphatase group by alkaline phosphatase. Okay? This is the beautiful enzyme which is often used in recombinant DNA technology experiments. Now, when to use CYP, that is calf intestinal phosphatase? It is often preferred over BAP because it is essentially denatured. It easily undergoes denaturization at 68 degrees Celsius in presence of SDS, that is sodium dodecyl sulfate. But if you want to denature on the other hand the BAP, you have to put it for a series of chloroform phenol extraction experiments, and this is comparatively a simpler procedure you simply can denature it at 68 degree Celsius in presence of SDS. That's the reason why SIP is preferred over BAP in recombinant DNA technology experiments. Then the reaction with SIP is usually carried out at 37 degrees Celsius, but if you want to carry out a catalyzing reaction using BAP, you have to provide 60 degrees Celsius. These are the two reasons why SIP is preferred over BAP. My dear students, by now I think you have understood about DNA ligases and also another nucleic acid modifying enzyme, that is alkaline phosphatase. So this is the quota for today's lecture. Thank you so much for your patient hearing. Stay blessed. Any queries, any doubts, do contact me over my cell phone. Thank you.